Hi everyone, welcome to this revision video about DNA replication. This video is based on the AQA specification A-level biology, specifically point 3.1.5.2. Let's get into it. First of all, let's get into some key definitions you need to know. Semi-conservative replication is when each strand is copied, so it acts as a template, so the daughter DNA contains one new strand and one parent original strand. Conservative replication, a DNA double helix is formed containing two new strands and none of the original DNA. Base stacking are the interactive forces between the base pairs that hold the DNA molecule together. Moving on, let's consider some terms. When we refer to a parent strand, we're just talking about the original strand. And when we talk about the semi-conservative model, we are also talking about the Watson-Crick model. Moving on, let's have a recap about DNA structure. As you saw in the video about structure of DNA and RNA, which I believe was spec point 3.1.5.1, we did talk more in depth about the structure of DNA, so make sure you hop onto that video if you just need a little recap on that. But just to recap, you can see, let me get my laser pointer out, you can see what we mentioned last time at the 3' end and then the 5' end. And I did spell this wrong, so let me add a quick T there. And a feature of the DNA structure is that sometimes it is referred to as anti-parallel, which means two DNA strands run in opposite directions. So as you can see, let me get a highlighter. As you can see, if we're looking at this new strand, it's first at the top, we're at the 5' end, and then it runs to the 3' end. Whereas if you look at the original strand, it starts from the 3' end to the 5' end. So it just runs in opposite directions. The first one goes from 5' to 3', whereas the other strand goes from 3' to 5'. Moving on, let me get back my laser pointer. Okay, some adaptations of DNA molecules. So DNA is double-stranded, and this helps because it provides stability. This is due to the sugar-phosphate backbone. There are many hydrogen bonds providing strength, and there's also base stacking, which we mentioned in the definitions. As it's double-stranded, this also protects the bases. This is due to the sugar-phosphate backbone. And also, as it's double-stranded, this helps replication, and it enables replication to occur semi-conservatively. DNA is a large such long, so it can carry lots of information. DNA has a helical structure, so it's compact. It contains the DNA base sequence, so it allows transcription. It contains a sugar phosphate backbone, so it protects bases, as mentioned before, and it contains weak hydrogen bonds, which is good for easy replication because they can easily be broken down. There are two models of DNA replication, the semi-conservative model and the conservative model. At least you need to know. They may introduce new models into the exam, but if you just consider the conservative model as like an opposite model, just to consider, just so you have an idea that there are other ideas that people had about DNA replication. So let's talk about the semi-conservative model. It was founded by two scientists called Watson and Crick, and to underline what the semi-conservative model actually is about, it is DNA helicase, which is an enzyme, unwinds the double helix structure of the DNA by catalyzing the reactions that break down the hydrogen bonds between complementary bases in polynucleotide strands. Both strands act as templates. Free DNA nucleotides are attracted to the exposed bases on the template strands and line up in complementary base pairs. DNA polymerase joins adjacent nucleotides of the new strands by catalyzing the condensation reactions that form phosphodiester bonds. Hydrogen bonds reform. Each new DNA molecule contains one old /parent /template strand and a new strand. And let's consider some roles within the semi-conservative model itself. So when you look at the enzyme, DNA helicase, it is used to unwind the double helix structure of DNA by catalyzing the reactions that break down the hydrogen bonds between complementary bases in the polynucleotide strands, so nucleotides are attracted/attached, or strands act as templates. And when we look at the enzyme DNA polymerase, oh my goodness, it joins adjacent nucleotides of the new strand by catalyzing the condensation reactions that form phosphodiester bonds / the sugar phosphate backbone. Moving on, let's consider the 3 DNA nucleotides that are added. These form complementary base pairs, adenine, thymine, cytosine, guanine, or form complementary DNA strand. They mean the same thing, so you can just pick either one. And when we consider the single-stranded DNA polynucleotides trains that we form at the start, they act as templates and they determine the order of the nucleotides that are added on, the 3 DNA nucleotides we mentioned above. Moving on, so if you were to compare the semi-conservative model and the conservative model, the semi-conservative model is the model used for DNA replication. It is universal in all organisms and ensures genetic continuity between generations of cells. This is very important, that last line. And the conservative model is not used as the model for DNA replication. So just to make that clear, the conservative model is not the one used for DNA replication. And as you can see, if you're watching the video on YouTube or Spotify, or if you're listening to the audio, make sure you hop onto YouTube so you can see these. There are quite decent diagrams of each of the models, which I think are really helpful, so make sure you look at them. And now we're moving on to the evidence for the Watson-Crick model, and this evidence was founded by the scientists Maselson and Stahl. I'm sorry if I said their names wrong. I hope I didn't. But yeah, those 2 scientists. And here's some key facts to know about the evidence used to justify the semi-conservative model. That all bases in DNA contain nitrogen, as they are called nitrogenous bases. They must contain nitrogen. Nitrogen has 2 isotopes. If you do A-level chemistry, you know about isotopes. But for just A-level biologists, I would just say it's not a direct comparison, so don't come for me for this, but just consider it like isomers. When we looked at the structure of glucose, how we saw there's alpha glucose and beta glucose, just consider it similar to that. And these isotopes are 14N, which is lighter, and 15N, which is heavier, and bacteria will incorporate nitrogen from their growth medium into any new DNA that they make. So moving on to this evidence for the model, 2 samples of bacteria were grown, one in light nitrogen, the other in heavy nitrogen. The bacteria gradually take up isotopes to grow. A sample of DNA was taken from each and spun in a centrifuge with heavy nitrogen DNA settling, forming a band lower than the lighter nitrogen DNA. A sample of bacteria that had previously been growing in the 15N isotope of nitrogen was placed in a 14 nitrogen growth medium. They were left for 1 round of replication, and a sample of its DNA was centrifuged. The DNA settled to form a band in the middle, showing that the bacteria contained a mix of the heavy and light nitrogen. This shows that the bacteria had semi-conservatively replicated. And with that, this revision video is done for DNA replication. Make sure to comment down below any suggestions of any other videos that you want to see. And if you need any more help on this topic, just make sure I know down below and I'll try and get to you as fast as possible. Thank you for watching and happy revision.